Welcome everybody again to Nephrology Grand Rounds, a virtual version today. Hope everyone's well and in the Valentine's Day spirit. Um, we're gonna have today first. We're gonna have our very own Kim Muczynski giving a talk, and then we'll move on next to our biopsy conference. So Kim is already loaded, has her slides up and visible. So Kim, if you'd like to get started, we're ready.
All right. Thanks, Daniel. Good morning, everyone. So my focus of research and nephrology has been on human kidney immunology. And I want to share a story with you today from my research that I think I have come to the conclusion that the kidney microvascular endothelium, in other words, the capillaries of the endothelium, confer a peripheral tolerance. And they do this by the class 2 molecules, HLA-DR being the main one. And I have some disclosures. One, I am not an immunologist. Immunologists deal with mice. I deal with humans. And the story I'm going to tell you has nothing to do with mice. It's human specific. So to start with, I'm going to go over a couple slides to talk about T cell and HLA basics, which you'll need to understand my research. I'm going to talk a little bit about toxicity of one of the checkpoint inhibitors, PD-1. I then want to make some comments about transplant and fetal development for T cells and some of the secondary signals. I'll give you my biological speculation on what I think my data means. And then I also want to introduce a couple co-stimulatory blockades we're going to be seeing clinically that I think are going to have kidney significance. So to begin with this background, T cell tolerance, it means that you are immunologically unresponsive to your T cells. If you look at any of the immunology textbooks, all of this is based on mice because you can manipulate mice far more so than you can humans. They divide tolerance into a central tolerance, meaning in the thymus, and in this nice, healthy young thymus, they select for T cells that recognize HLA. And then if those HLA molecules see an antigen that's being expressed in the thymus, it's deleted. So for instance, when Michael Tash, one of our fellows, was with me, he found nephrin in the young thymuses, suggesting that any T cell would have been deleted that recognized nephrin. Now, what happens when you get old is there's involution of this thymus, so you don't have the same sort of robust ability to delete T cells. And the T cells, which are sort of naive and young in the young thymus, become old when you look at them in the adults. So what happens when this thymus involutes? And does this mean that you are going to get autoreactive T cells? And what happens if these self-reactive T cells escape the thymic deletion? We have a backup system for tolerance, and that is something that happens outside the thymus, and we refer to it sort of as anergy. The idea being that you have mature T cells that are exposed to antigen, but they don't respond because perhaps they don't have co-stimulation, they're being inhibited, or there's some regulatory T cells. But there is a peripheral mechanism for making us so we don't respond to autoantigens. And understanding how this energy occurs is really key if you want to harvest it to turn off T cells in autoimmune conditions or transplants, or to increase the T cell killing on cancer cells. And manipulating energy is a clinical reality. We all know that. There are these checkpoint inhibitors, which are primarily antibodies to program death protein and its ligands, and they're used to block the anergic T cells so that you can then kill your cancer cells. Approved a decade ago, and there's more in the pipeline, which I'll mention at the end. Now, how does a T cell recognize an antigen? Well, they talk about signal one, and signal one means that the antigen presenting cell has an HLA molecule on it that binds the peptide, and that peptide bound to the HLA molecule is sensed or is seen by a T cell receptor, allowing the T cell to proliferate, make cytokines, do whatever. If you do not have a peptide, you don't get a response. If you have an HLA molecule that's of an allele that won't recognize the peptide, you don't get a response. And if you don't have any kind of HLA molecule, there's no response. Now, the HLA molecules are of two classes. Class 1, when you're looking at HLA reports and transplant, there are A, B, and C alleles, and there are HLA class 1 molecules are on all nucleated cells, and they present their peptide and antigen to CD8 cytotoxic cells. There's another class of HLA molecules, class 2, which are represented by DP, R, and DQ, and they are constitutively on in B cells and dendritic cells, but they can also be induced in other cells. So class 1's on all nucleated cells. Class 2 is restricted just to B cells and dendritic cells. So the literature says about mice. And they present their antigenic peptide to CD4 cells. And this is the focus of my research, is this class 2 molecules. And I'm going to be talking primarily about DR, which is the most abundant. Now, you need a signal too in order to trigger a T cell. So this HLA peptide T cell, it's very specific, but it's weak. It needs some other receptors in order to trigger it. So there's a bunch of proteins that can cause co-stimulation. On the other hand, there's also a series of proteins that can inhibit a T cell response. And whether or not this T cell is going to do its thing usually has to do with the phosphorylation of the T cell receptor, with co-stimulation phosphorylating this and some of the inhibitory molecules taking the phosphates off. Now, where does this peptide here, where does that peptide come from? So if you are a class 1 molecule, this peptide is coming from an intracellular protein. So the idea is the cells get infected, there's a virus, or they transform to a tumor, and that peptide then is presented by the HLA class 1 to the CD8 cells. Class 2 molecules are a lot different. What they do is they take up extracellular proteins, they endocytose proteins, whatever's going by, and then they can bring it into little bits of peptide and present it on the class 2 molecule to the CD4 T cells. So with the class 1 molecules, they're presenting our intracellular peptides. Makes sense. You kind of want to kill those cells that are infected or becoming tumors. And in the class 2 molecules, they have this ability to sample the environment around and then to present that peptide. So that's the basics. So let's talk about my research a bit. And the story begins with Bill Kauser. And Bill Kauser and I had a very good relationship. So I told Bill that I had no desire to work for anybody in his division when it was time for my research. And Bill was very good about this. And he actually encouraged fellows to go out to bring some science that could be then applied to nephrology. So he had no problem when I found the lab of Don Pius, a pediatrician, which I joined in 1990, probably would have still been there, but unfortunately Don developed pancreatic cancer and died during my fellowship. That aside, Don was very good in his lab about developing some genetic mutations in order to study how you regulate those HLA class 2 genes and proteins, and to determine how those antigens actually fit into HLA molecules. And all of the work that he was doing was all human, no mice involved. And there's where I met Susan Anderson, an extraordinary research scientist and microscopist who had actually worked for somebody by the name of Andy Farr in biostructures here. And Andy had studied mice kidneys and had found that there were HLA molecules in the mice kidney, but they were on macrophages. So Susan just kept pushing and pushing, and she said, go get me some human kidney because I want to see where the HLA molecules are. So I went out and found some normal human kidney, and she put on an antibody that would recognize all of the HLA molecules, not just any one specific allele. And what we found is that HLA was highly expressed on these cells within the interstitium and in the glomerulus and sort of in an endothelial pattern, but they weren't on cells of larger vessels. And to prove this, then we took another antibody to endothelial cells and showed colocalization of the HLA-DR and the endothelial marker. And so what this shows is that this HLA-DR is expressed at high levels on these kidney microvascular endothelial cells, Kmac I'll call them here, but not on the endothelial cells of larger vessels. And throughout this talk, when I'm referring to endothelial cells, just to shorten it, I'm referring to these capillaries, the microvascular endothelium. Now, this was a concern for me. I thought for years this was all wrong, that I had screwed something up. And it was a concern because if these cells in the kidney, these endothelial cells, are expressing this HLA-DR, this class 2 molecule, it means they have the potential for presenting proteins or peptides from the circulation to activate T cells. But I'm looking at normal human kidney here, and there's no inflammation. And yet these endothelial cells in the kidney are constantly being bathed with proteins that they can actually take up and process to activate T cells. And I worked really hard to get fresh kidney tissue. So I used to go to the operating room, still do, and get the kidney from nephrectomies when it was still warm to make sure there was no manipulation being done with the tissue. But then I also realized that I was not the first to make this observation. It was Charlie Alpers here within our pathology department. And Charlie actually went looking for macrophages in human kidneys. And what he came up with the same thing. He was looking for macrophages using the class 2 antibodies, and he found an endothelial pattern. And there's also an investigator from the UK who's done extensive work in heart and kidney that showed a similar observation. So these normal human microvascular endothelial cells have high expression of this HLA-DR, another class 2 on their surface, where they can encounter circulating T cells and potentially present a peptide, but there is no inflammation. So why is this? Maybe it's because all those autoreactive T cells are deleted in the thymus. The thymus is so good that even now at my age, I'm not getting autoimmune disease because I had a very active thymus. So there's just no T cells to see those peptides. Or maybe there's something odd about the regulation of how you get HLA-DR on these cells. Or maybe the endothelial cells can't really present peptide to T cells. Or maybe they don't have this secondary co-stimulation. Or perhaps they have inhibitory signals that prevent the inflammation. So in order to answer some of these questions, I had to develop a technique where I could take kidney tissue from biopsies or from my nephrectomy samples and turn it into individual cells. Take all of this, make it all individual cells without losing the epitopes, the proteins that I wanted to look for. And it took me a couple years to do this, but I was able to do that. And so I was able to isolate all these kidney cells. I also was able to isolate cytokines, which we won't talk about today, that you can capture on beads and measure. And with the cells, then I could add antibodies to the surface protein, each conjugated to a different fluorescent molecule, do something called flow cytometry, where I could identify the cell types and their immune markers. So now I had a technique where I could actually take kidney tissue, go to individual cells, and what you're looking at here is tissue that has gone through my dissection, essentially, and each of these dots is a cell. And what this does is it allows me to analyze the marker on these cells, or I can sort these cells out and isolate them for culture. So here's my microvascular endothelial cells that have high expression of DR and the endothelial marker. The larger vessels do not express DR, and that they do have the endothelial marker. And then if you want to separate out, say within this microvascular cells, you can either tell them whether or not they're peritubular capillary cells or they're glomerular capillary cells by using some other markers. But the key thing is I now had a way of studying some of these cells. So one of the first things I wanted to do was to look at the regulation of these endothelial HLA-DR. And it turns out that class 2 genes are expressed whenever there is this, they call it a master regulator, C2TA. When C2TA is turned on in cells, then the cells express class 2 genes. And what C2TA does is it binds to a lot of DNA binding proteins. It itself does not bind to DNA. But what's interesting about this master regulator of C2TA is it has 3 different promoters. So dendritic cells and B cells that are always expressing class 2 have promoters they use. And then for cells that can be turned on to express C2TA, there's a gamma interferon promoter. And so when I looked at my endothelial cells, I found that the C2TA is definitely present. It uses this promoter, but it only requires a physiological concentration of gamma interferon, which is why you see it in normal kidney tissue. You don't need to have an inflammatory response to see this HLA-DR. Go back here to this signal 1, where remember a T cell has to have a HLA molecule presenting a peptide to a T cell receptor. I wanted to see if I could make my endothelial cells actually present peptide to T cells. So I worked with Jane Buckner at the Benaroya Research at Virginia Mason, and she provided me some T cells that were specific for recognizing an influenza hemagglutinin peptide when it's presented by this particular DR allele 0401. And so I I went ahead and to see if I could get my endothelial cells to activate T cells. I compared endothelial cells that had the right HLA-DR type with ones that did not. And I also used B cells sort of as a positive control with the right HLA-DR type. And what we're looking at here is just T cell proliferation based on thymidine uptake. And if I don't put anything into my endothelial cells or B cells, this is a baseline function. If I add peptide, it doesn't change much. If I add T cells, it doesn't change much. But when you add peptide and the T cell to the right HLA type, you get proliferation. B cells proliferate a lot more. If I have an HLA type that does not recognize the peptide, there is no proliferation. So these endothelial cells will cause proliferation of specific T cells with peptide when it's presented by the appropriate DR restriction. But I also said there's got to be a signal too that's going on when T cells turn on. And so I wanted to go ahead and look for that signal. And so I continued on with some of these T cell assays using some endothelial cells I had isolated. And here again, we have my endothelial cell. Here with peptide and T cells, I've got a proliferation. But if I block the DR with an antibody, I don't get proliferation. If I block something called CD58, leukocyte function antigen, I don't get proliferation. And look what happens when I threw in this antibody that some investigator gave me. And at the time, I don't think anybody really knew what it was. It was an antibody to a program death ligand. I got more proliferation. And if you don't like to do proliferation assays, you can also look at the interferon produced. And again, with this antibody to a program death ligand, I had more activity. So that these endothelial cells, the activation of T cells requires DR, it requires CD58, and if I block this program death pathway, I get enhanced activation. So if I go back to the questions that I had asked, I've now been able to answer somewhat with the isolated endothelial cells. I don't see inflammation in the kidney. Maybe still it's because there's no T cells to see HLA-DR peptides, but I think that's unlikely. The HLA-DR is regulated by C2TA. The endothelial cells are able to present peptide to T cells. They don't lack secondary signals. They actually have it, CD58, and they do have some inhibitory signals. So maybe it's these inhibitory signals that are keeping the T cells quiescent in the kidney. So I then started working with Elena, who is now my pro at flow cytometry with me. We laugh, we can do one experiment and analyze it for probably years. And what I wanted to do is to summarize is in the endothelial cells, what activating signals are present and what inhibitory signals are present. And at the same time, I looked at the kidney T cells, not T cells in the blood, but the T cells that are actually in the kidney to see if they have ligands for these activating signals or these inhibitory signals. And these molecules have been reported in different systems to activate or inhibit T cells. If there's an open thing here, it means I evaluated it, but I didn't find it in either the endothelial cells or the T cells. And if it's shaded, it means it was present. These dot blots are just my flow cytometry to show you that the signal is there. And I'm looking here at normal native kidneys, and this diagram probably summarizes, I don't know, 10, 20 kidneys that I've looked at over the years. So in those isolated T cell assays, I block CD58 right here. And indeed, CD58 is on the endothelial cells. It's got a very high level of expression, binds to CD2, and indeed, CD2 is on the T cells. So blocking this meant I could turn the T cells off. If I looked at the endothelial cells for these program death ligands, here you have it here, the program death ligand 1 and 2, both of which are on in this kidney, and you have the receptor program death on the kidney T cells. It's not on all the T cells, and depending on your donor, it may be in as many as 70 or 30%, but much higher levels of this program death receptor in T cells in the kidney than just in the circulating blood. And when I had blocked this, that's where I got even more proliferation of the T cells. So what I really wanted to do is to check this whole thing out in humans. And so the human experiment is, can I block this PD-1 pathway? And I was fortunate that the oncologists later started to use these checkpoint inhibitors, most of which block this programmed death pathway. And what we have found over the years, and we started off, that an acute kidney injury can occur with these. No, Volamab is one that was early on, and we don't even biopsy these kidneys anymore because it's fairly common. But for patients who had some metastatic tumors, they developed acute interstitial nephritis, was mononuclear, eosinophils weren't present, the gloms look pretty normal, there's no immune complexes. There's other organ toxicities that form, and I propose that this interstitial inflammation is because of the recognition of the microvascular endothelial HLA class II by T cells without this inhibitory signal of programmed death 1. And so the toxicity of these checkpoint inhibitors actually confirmed some of my vitro data. And if you want to know what are the infiltrating cells, what would you expect? Primarily T cells, and they do predominate when you have the interstitial nephritis from checkpoint inhibitors. So if we go back here, what I'm saying is that there are a couple of checkpoint inhibitors, pembrolizumab, nivolumab, and they actually block this program death 1. And so by blocking this signal, what happens is that you can be at risk for getting an interstitial nephritis if you have an endothelial cell that has this signal 1 where the T cell can recognize. You got to have this signal. This is why not everybody with checkpoint inhibitors gets toxicity. The endothelium has to recognize a T cell, but if you block this inhibition, you're set up for getting inflammation. Just about the program death, which I mentioned again, T cells respond because they get phosphorylated, and the program deaths take off those phosphorylations, and that's sort of the mechanism and how this works. So just in summary at this point, the microvascular endothelial cells of the kidney express HLA class II, which allows them to sample all that extracellular environment, the protein debris that's going by. They can activate T cells in a DR-restricted manner, depend on peptide, but T cell activation is limited by this program death 1 pathway. And so my conclusion is that we have a peripheral T cell tolerance system based on kidney microvascular endothelial cells expressing DR, but it does it in the setting of a programmed death receptor so that it allows us to be tolerant of some of those proteins that might cause autoimmune disease. Now, I used to think that the kidney was very special because it had a lot of filtrate going through, but it seems that there are other organs where HLA-DR expression has been found on microvascular endothelial cells. There's some similar findings in the heart and the gut, but none of them have really been evaluated in any detail. The literature on this is old, but it also raises the question of whether or not humans have this microvascular endothelial cell with HLA expression that causes anergy, and that it might be based on capillary beds throughout multiple organs within the body. I want to make a couple comments real quick about transplant. And when I have looked, thanks to Nikolai, and we probably have 50 transplant biopsies that Elena and I have looked at, the endothelial cells have similar amounts of DR, similar amounts of the protein death ligand, with or without rejection. If you look at the T cells in transplant biopsies, they have this programmed death. One with or without rejection. There may be more infiltrating T cells, but they don't necessarily have more programmed death on it. So it suggests that these inhibitory signals are probably insufficient to totally prevent rejection. But if you block these signals, you can get rejection. There's a very interesting paper that came out in a kidney transplant recipient treated with a checkpoint inhibitor for his melanoma. And what they looked at was the T cells within his kidney. And they found that they were clonally expanded, suggesting that not just any T cell is in the kidney, but an alloreactive T cell is present in the kidney that when you remove the inhibitory signals allows it to expand. So when you're talking about transplant or autoimmunity versus oncology, they have opposite goals. Cancer, you want to increase your T cell activity. get rid of this inhibition. In transplant, you want to decrease the activation and increase the inhibition. And indeed, that's now what's being done. There are some protein death 1 antibodies that recognize proximal area of the protein, and they're agonists. In other words, they increase this inhibition. And that's something that we'll see later on. But the real goal is you want to be more specific about what T cells you're activating or inhibiting. And there are now some things that are trying to target part of the T cell receptor in a bifunctional agent that would also inhibit the T cells. But I think what we really need is a T cell specific bifunctional agent that recognizes the HLA, the peptide, and then says, turn that T cell off. And this is actually being worked on. And if you think about the David Baker's lab and the Nobel Prize, we have actually probably the expertise within our university to start thinking about some of these uh, bifunctional agents. Finally, a couple comments about fetal tissue. We are fortunate here that we have this birth defects research lab in pediatrics that can provide de-identified fetal tissue, and this is again human. And so I asked, when is the DR expressed on the microvascular? And indeed, by about 60 days gestation, the endothelium in the fetus expresses DR. And does it have the ligands for this program death 1? Yes, these ligands are present here. And so what it suggests to me is that this expression of DR that you're seeing in the fetus with the ligands on the microvasculature early in development supports that there probably is an endothelial-based immune system. It's not induced by inflammation. It's present in all of us at an early stage. Biological speculation. Why would you want secondary signals on the endothelium that will both stimulate and inhibit T cells? And it turns out that this inhibitory signal program death 1 is not present on naive cells. It appears only after the T cell has been activated and have a little activation trickle to the T cells that recognize our autoimmune HLA peptides so that you can turn on the inhibitory signals, a small activation that can be turned off rather than having a massive inflammatory response. And what about the significance of this microvasculature early in development? The human thymus develops about day 38. T cells appear about 60 days, which is when you see these microvascular endothelial cells with HLA-DR. If you do a neonatal thymectomy, which happens in cardiac surgery for congenital problems, the kids still grow up and be healthy adults. If you do this in mice, they get massive lymphocytic infiltrates. So perhaps in humans, we actually need more of this peripheral tolerance system to take care of our autoreactive T cells that escape the thymic deletion. And we just don't appreciate it because all the textbooks just talk about mice. And when I think about this, maybe what we're trying to do with this endothelial barrier is prevent developing parenchymal cells. So the endothelium can sort of take up any debris that might be recognized by T cells and has developed a way to make it anergic. And just in the last 2 minutes, I want to tell you about 2 other things to look for. We're having some other oncology agents that are going to be created. One is to the CD276, which is present in my endothelial cells, or also known as B7H3. They don't know the receptor, but one of the putative receptors is phospholipase A2 receptor. So I'm wondering as blockade comes out here, if we're going to start seeing something like a membranous lesion or something else. And these agents are actually out there. Some studies have been completed. One is working on a bifunctional agent to specifically target the T cells. And there's another one that's quite interesting. The CD151, which provides the poliovirus receptor, has 2 ligands, one that will activate a T cell, CD226, and one that turns it off, something called TIGIT. And so when cells get activated, TIGIT comes on to control that. And there are now efforts to try to block this CD151 here. And they're recruiting studies right now, usually people with have fairly extensive cancers, but they are also trying to make CAR T cells to CD155. And what's interesting is that when you read these papers, which are quite new, CD155 is said to be selectively expressed in cancer cells, rarely in normal cells. And yet I look by my flow cytometry, All of the endothelium in the kidney has high levels of CD155, so I look for more toxicity there. So the final conclusion is that I think kidney endothelial cells, the microvascular ones, via their HLA class II, create an immune system by which we have peripheral T cell tolerance. And it does that by these programmed death ligand and other inhibitory signals. So the kidney is more than just a filter to take care of our electrolytes and our volume. It also contributes to our immunity. And these are the folks that have been instrumental in this work. And I'll thank you for listening. And I might have 1 minute for a thought or put them in the chat as we go on to pathology.
Well, thank you so much for that, Kim. That was really amazing work that you've been doing, and um, oh, very nice. Is that from one of your adventures?
Ah, his name is Orion. Orion was my friend for several days.
Well, very nice. Well, thank you again, and we maybe have time for maybe one question or comment um, before we move on to pathology. If not, we can certainly continue in the chat as well. Yep, Bessie.
Hey, Kim, great talk. Thank you. I may have missed it, but is there something that we can give when people are getting the checkpoint inhibitors to sort of inhibit the response?
No, I don't think so. But we could do is screen for it. In other words, you could see if they have T cells that will recognize things presented by the HLA and probably just with, the proteins that are present in the blood. So you could develop a whole assay system to probably
Mhm.
predict this.
Okay, thank you.
All right. Well, thank you so much. I think just for the sake of time, we may move forward with pathology conference, but please send any additional questions or comments in the chat.